Welcome back, everyone. Our last presenter is Dr. Nasir Mirza. Dr. Nasir Mirza is a senior clinical lecturer at the University of Liverpool's Institute of Systems, Molecular and Integrative Biology. Dr. Mirza's clinical interest is in epilepsy, and his research interests lie in using genetic and genomic data to accelerate the discovery of better treatments for epilepsy. Dr. Nasir Mirza is using Sysrev to create the computationally aided curation by hand of epilepsy proteins. I say CACHEP, Tom says CACHEP, we're not sure, a database of the genes and proteins underlying epilepsy across syndromes and species. Take it away, Dr. Mirza.
Great. Hi, everyone. Thank you very much for asking me to talk about the CACHEP project. So what is the CACHEP project? As you just heard, the CACHEP stands for Computationally Aided Curation by Hand of Epilepsy Proteins. And the goal of the project is to create a database of epilepsy genes and proteins. And in this presentation, the terms gene and protein are used interchangeably. So why make an epilepsy gene database? So there is an urgent need to find better treatments for epilepsy. So each of you knows at least 6 people with epilepsy, although you might not be aware of this. A diagnosis of epilepsy is often not disclosed. Of the 6 people with epilepsy that you know, 2 people continue to have frequent recurrent seizures despite taking anti-epileptic drugs. So 1/3 of people with epilepsy continue to have frequent recurrent seizures despite taking maximum doses of multiple anti-epileptic drugs. Of course, this has a huge adverse impact on them, their families, on society in general. So there is an urgent need to find better anti-epileptic drugs. Hundreds of different genes have been found to harbor mutations in people with different types of epilepsy. Similarly, the manipulation of hundreds of different genes has been found to cause epilepsy in animal models. By collating and integrating this information, the genes and networks of genes that have the strongest evidence of causing seizures in the widest range of patient populations can be identified. These genes can then be targeted using existing or novel compounds. So the aim of the CACHEP project is to create a literature-derived database of genes that underlie seizures in different types of epilepsy in different animal species. An additional important aim is to promote interest and participation in science and research among students by inviting and enabling them to take part in this project. So this is the method we adopted for this project. So a literature search was performed for abstracts mentioning terms related to epilepsy or seizures and gene mutations or manipulation. Students read the abstracts in order to identify relevant papers and extract the required information from them. We mobilized and organized a large number of students to perform this large-scale data gathering project. The database itself is near completion, but not quite complete, so I won't be talking about the database itself, but I will talk about how we mobilized and organized a large group of students for this project. And I hope that our experience will be useful to other people who want to perform similar projects in the future. So I will be talking about the task that we were tackling, how we recruited a team to tackle this task, how we decided on the time period and the terms and conditions of tackling this task, and our training and audit processes. So our task was to to read and extract data from nearly 10,000 abstracts. And each abstract had to be read by at least 2 different people, and at least 2 different people had to extract data from them. So it was a huge task. How did we recruit the members? So more than 60 students contributed to the project. About a quarter of the students were high school students. So in the US, I believe you'd call it 12th grade. We call it 6th form. We invited students from different types of schools. We wanted to see if there was any difference in the levels of participation of different types of schools. So we invited students from a normal standard school, non-fee paying, from a specialist life sciences school, also non-fee paying, and thirdly from a private fee-paying school. The rest of the students were undergraduate medicine or biomedical sciences students. We had a focus group of 8 members, and we had an admin team of 4 members. So the 12th graders were recruited by contacting the school administrators directly. The undergrads were recruited by word of mouth, and emails were circulated to year or course groups and to research or neuroscience student societies. We wanted to ensure that potential participants were fully informed about what they were signing up for before they decided to join. So we wrote and circulated a guidance booklet and created and circulated a short YouTube video, and we asked them to look at these carefully before they signed up. Time period. So participants were asked to start and complete over the Christmas and New Year holidays. We envisaged that this was an opportunity to achieve something worthwhile during a Christmas season which was subdued by the pandemic lockdown. Our focus group agreed, and our student researchers also felt the same. So we are very keen to ensure that the students who did the research become co-authors on the publication that will hopefully ensue. In the interest of fairness and to avoid gift authorship, we wanted to ensure that everybody contributed equally. So we decided on a minimum number of abstracts to qualify for authorship. To decide the minimum number of abstracts, we did dry runs using our focus group, and we estimated the time needed to read each abstract and extract data from it. And we decided on a number of abstracts which would equate to a significant contribution but would still be manageable. The task had to be completed by the deadline, and we also stipulated that if there was significant discordance for any student researcher, that they would be excluded from authorship, although this never ended up being a problem, thankfully. So the students did have to decipher advanced genetic and neuroscience terms and concepts which they weren't familiar with before. So a detailed guidance booklet with illustrations and examples was prepared and circulated. The 12th graders were also provided with live tutorials. And the admin team performed live audits of the initial responses from student researchers and provided individualized feedback. 90% of the students completed the tasks, the 12th graders and the undergraduates. The whole data gathering exercise was completed ahead of schedule, and there was no evidence of a systemic difference between the quality of responses from the 12th graders and the undergrads. We asked for feedback from the student researchers. So this was the feedback that we got from the 12th graders. So we asked them about their motivation for taking part in the project. Unsurprisingly, they were already interested in science-related subjects. Some felt that taking part would give them an advantage when they applied for medicine courses, to study medicine or for other courses. Some were interested in learning more about epilepsy and some about genetics. These are the kind of benefits that they felt that they obtained. All of them felt that they had a better understanding of epilepsy. 80% felt that they gained a better understanding of genetics, and all felt that it would help their university applications. These are some of the qualitative responses that we got from the students. They felt that they had a better understanding of the different types of epilepsy and the clinical features of epilepsy. They felt that they gained an insight into research, into analyzing data, and working as a part of a team and to a deadline. And it was very gratifying to see that many of them felt that taking part in the project increased their interest in doing research in the future. This is the feedback we got from the undergrads. So their motivations included being interested in pursuing neurology as a specialty in the future, or to improve their skills, their knowledge, or to improve the CVs. 90% felt that they gained a better understanding of epilepsy having taken part in the project. 90% felt that they gained better understanding of genetics. And this is some of the qualitative feedback that they provided. They felt that they gained skills in systematic review, in collaborative working, in time management, in reading scientific literature. And again, it was gratifying to see that uh, many of them felt that they would be more interested in doing research in the future, having experienced this research project. So in terms of the progress with the database itself, the post-processing is nearly complete, including resolving conflicting answers, mapping and harmonizing gene annotations, and addition of data from other resources. And it is hoped that the database will be uploaded and submitted in the near future. So these were some of the learning points for us from this project. There's a high level of interest and enthusiasm among students for participating in such research projects. Students of different ages, experience, and backgrounds were able to complete the research project successfully. We did find that the proportion of students who volunteered was relatively lower from the non-fee paying schools. So for future projects, we will undertake additional engagement and outreach with those types of schools. We found that a short, sharp burst of intense activity was very effective for this kind of project. But choosing the right time period is key. We chose to do it over Christmas during a pandemic lockdown. Um, but what has to be borne in mind that such a project would be difficult to do in normal term times, but perhaps outside term times, it should be certainly possible. Having a focus group was very beneficial for us. And of course, having an eager and efficient admin team was absolutely essential. Speaking of which, a huge thanks go to Lara, Dan, and Basel, who are student researchers, but who are part of the admin team and worked very, very hard and very efficiently to make this project possible. And of course, thanks to all the student researchers who have taken part. Thank you.
Thank you, Dr. Mirza. I know I can speak for Tom when I say that hearing about the educational benefits of Sysrev, while it wasn't the first thing we were trying to accomplish, has definitely been one of the greatest joys to see that be, again, an unintended consequence. So thank you for that presentation. Everyone else, the panel will be beginning shortly, so don't head too far. Thanks again, Dr. Mirza.
Thank you.